Hi there, peace be upon you. Today I'd like to present a project that I have completed in my master's thesis. So my project is entitled as Synthesis Antimicrobial Evaluation, Docking and Dynamic Investigation of Novel Stearoyl Mannopyranosides. In this specific project we have synthesized a compound, and then we have done acylation reaction and lots of things. But at the first step of our research project, we have uh, choose a compound. The compound was basically methyl alpha D mannopyranoside. That was our starting material. So you are seeing that number on label compound that is called, you can say, methyl alpha D mannopyranoside. Then we have done acylation reaction in standard condition, and we have used different acylating agent for produce different derivatives of methyl alpha D mannopyranoside. And when we have doing this project, our main objectives was basically synthesize a series of novel sterile mannopyranocid for effective antimicrobial agents, and the next one, evaluate the antimicrobial activity of these synthesized compounds through SAR molecular docking, pass studies, molecular dynamic simulation, and admit studies we have also done. Actually, in the first step, we have completed, we have chosen the compound on, and we have done molecular acylation reaction using different kinds of acylating agent. But at the first step, we have chosen stearoyl chloride as a acylating agent, and pyridine that is used for making the basic medium, and DMAP that acts as a, you can say, you can say, catalyst to uh, make the reaction faster. And in this way we synthesize compound 2, and compound 2 again further acylate it using different acylating agent like octanyl chloride, decanyl chloride, dodecanyl chloride, and hexadecanyl chloride. And we have uh, produced or synthesized these derivatives. And we our main expectation or main objectives was to find out their drug likeness properties. Is any kind of drug likeness properties exist in these compounds or not? And at first we have done computational analysis that is called, you can say, pass analysis. Pass means, you can say, probability of activity spectra of substance. And from this specific portion we have find out, that means different pa and pi value. And from pa and pi value we are find out, that means uh, compound on or compound to have antibacterial, antifungal, or anticarcinogenic or antiviral properties or not. And from for referencing we have chosen some, you can say, standard you can say drugs like fluconazole, itraconazole, that was the antifungal drugs, and nystatin and tetracycline, that was the, you can say, anti bacterial standard drugs for our project. And when a compound that showed, you can say, higher pa value, means pa means probability to be active, pi means probability to be inactive. That means when the pa value is greater than zero point 0.7, that indicates higher probability to be activity. So that's our, you can say, initial approach. And then when we have done, you can say, good pa value in case of compound 3, we have found, you can say, compound 3, 4, 5, 6, we have found 0.673, that is tends to zero point 0.7. And in case of, you can say, compound 2, we have found, you can say, 0.769, that was the, you can say, highest pa value. And so compound 2 that has, you can say, anti-carcinogenic properties, and compound on that have, you can say, uh, you can say, pa value is 0.628. And at the same time we are seeing our standard compounds that showed, you can say, each and every standard you can say drugs that showed, you can say, larger you can say pa value that is greater than zero point 0.7. And from this specific you can say approach, we have found some, you can say, some initial uh, strategy to go through the you can say in the next steps. And we have completed molecular docking using pyrex, and some compound that showed some higher binding affinity uh, in kilocalorie per mole unit. And actually we have chosen some, you can say, fungal protein as a macromolecule, and we have chosen our synthesis compound as a ligand, and we have done molecular docking using pyrex, and at the same time head dog, and we have found different kinds of values, but some compounds that shows highest binding affinity. When we have found some highest binding affinity, we have approached in the next steps to do molecular dynamic simulation, so that we can find out, that means it is the complex between bacterial protein uh, against our synthesis compounds. If it show the highest stability, that means it is capable to inhibit the bacterial growth or fungal growth. So that means in such cases, we can say that our compounds uh, can act as a, you can say, probable drug for those, you can say, bacterial or fungal protein, and it can replace the existing standard drugs that we chosen in our research, like fluconazole, or you can say itraconazole, or tetracycline, that we choose as a standard drugs in our research. So that's the basically approach that we completed. And you are seeing the different binding affinity. Our standard drugs that shows the highest binding affinity, but our compound 2 that showed, you can say, uh, uh, that is a, you can say, good binding affinity, -7.7, that is tends to that of iconazole, or you can say ketoconazole. At the same time, compound 3 that showed, you can say, highest binding affinity among our synthesis compounds. So that is a, you can say, positive sign for us. And we have done molecular docking. We have found different kinds of parameters from molecular dynamic simulation. That means from the, you can say, graphs, like graphs of RMSD, root mean square deviation, root mean square fluctuation, that means RMSF, and radius of gyration. We have concluded that actually our compound that is stable, uh, you can say, bacterial or fungal protein, that indicates that our drugs that has capability to inhibit the bacterial growth or fungal growth. So that's our main, you can say, uh, success that we have done in this project. So that's all today. Thank you for watching this or listening overall description. Thank you.